In the NCRI 2020 meeting, we discussed our findings on the immune responses within pancreatic cancer, and specifically the T-cell responses. So as many of the audience are aware, pancreatic cancer itself is a deadly disease with a handful of limited treatment options. And checkpoint immunotherapy recently has heralded success in many different types of cancers, but yet we've failed to see a durable response within pancreatic cancer itself. So me and my colleagues and other labs around the world have shown that patients with T cells in their pancreatic tumors have a better survival. So what we've done in this piece of work is take the opportunity to actually characterize what those T cells within pancreatic cancer actually were. So just to summarize those findings, we've actually found that over half the CD8 T cells were actually senescent and effectively inactive and lying dormant. We've also found that there was a small population of cells as well that were activated and exhausted, but did not have high levels of PD-1, suggesting to us that PD-1 therapy may not work in this cancer in the naive state. We also looked at CD4 T cells, and we also found there that there was a significant population of senescent cells as well. But what was interesting is there was also a significant regulatory T cell compartment. And within that Treg compartment, we found that they were highly activated and suppressive. And they were heralded by the checkpoints ICOS and TIGIT. And within our work, we've also seen that TIGIT is another checkpoint on activated CD8s and also on NK cells as well. And we've also demonstrated in this piece of work that the CD8s and CD4s pretty much lie homogeneously throughout the tumor, whilst actually the regulatory T cells are pretty much exclusively in the stroma of this cancer. So this has actually led us to believe that in the treatment-naive setting, that ICOS and TIGIT inhibition should be investigated, and there are early phase trials of these compounds going.